everyone, today we're going to have a look at mass transport in plants, which is xylem and transpiration. This is a lesson which will go in two parts. This is the first part, the next bit will be about phloem and translocation. So today we need to look at a few things. We need to be able to describe the movement of water into the root, the process of transpiration through the plant, and then the cohesion tension theory of water movement. The first thing that we need to look at is how water potential is established in a gradient across the plant tissue. We're going to look in the leaves first, and we notice that the leaves have very low water potential because water is always evaporating through the stomata when they're open. In the roots, it's the opposite. They have a much higher water potential because they're going to be taking the water out of the soil through osmosis. Before we move any further, what I'd like you to do is pause to brainstorm key words that relate to the movement of water between two areas. So that's our process of osmosis. Give a lovely definition, please. Let's look at our definition first. Osmosis is the movement of water molecules down a water potential gradient. This occurs through a partially permeable membrane from an area of a high water potential to an area of a low water potential. So let's have a look at our roots on a plant. You can see there's lots and lots of fine hairs, fine root hairs, that will help the water move in from the soil to the roots so it can enter the xylem. Let's take a moment to pause and think about how these root hair cells are adapted for the rollers exchange surfaces for plants. First, you need to remember the three fixed laws, and then think about how this is a good adaptation for them. So the first fixed law we can look at is the fact it has a large surface area. The second is that there's a shorter diffusion distance. And the third is the movement of media to maintain a constant concentration gradient. Looking at the surface area first, root hairs are present and they have very long extensions on each root. If I go back to our picture, we can see that super clearly. So having each of these projections is going to help increase the surface area for absorption of water. The next one, to maintain a short diffusion distance, is that there is a thin cell wall on each root and root hair to reduce the diffusion distance. And lastly, to maintain a concentration gradient, we're going to be actively transporting dissolved sugars, amino acids, nitrates into the xylem to maintain a water potential gradient. So let's look at how water enters a root hair cell. And here we can see a picture over here where we've got water, we see the arrows to show it moving in, you've got the root hair, you've got the main cell, and you've got these soil particles all around. So the first thing that we want to think of is how we establish a water potential gradient that favours drawing water inside, because we don't have little water pumps here moving things for us. So in a root, they're going to absorb minerals such as nitrates and phosphates first through active transport. And we know that if you have high in the concentration of solutes inside a cell, this is going to lower the water potential inside the cell. Now we have a higher water potential outside of the cell where the soil is. And water is going to move down the water potential gradient due to this concentration, not due to pumping water from the outside environment inside the cell. So let's look at two pathways that water will now use in order to move its way from the root into the xylem. There's two ways we're going to focus on here. The first is called the apoplastic pathway, and we have the symplastic pathway. And one travels through the cell wall, whereas one is focused more on movement of water through the cytoplasm. Let's look at the apoplastic pathway first. Here you've got three cells lined up next to each other, and let's imagine that the outside of our root is over on the left, and that the right hand side shows almost close up to the xylem. What we can see is that these arrows represent where the water is travelling through. So the water starts off entering the cell wall here, where it has lots and lots of different structures inside the cell wall. We're going to have a look at the cell wall structure in a lot more detail later, that allows little spaces to be present, and those spaces are an excellent channel for water to pass through. So water moves through the the cell wall of these cells across the root cortex until you hit this layer called a Kasparian strip. The Symplastic Pathway is where the water wasn't moving in the other diagram. They're opposites. So in the apoplastic we move through the cell wall, in the symplastic we're going to move the water through the cell membrane, and you can see that there's this little joining feature between those two cells which is called a plasmodesma. And this allows the water to flow continuously from one cell to the next. So we're going to go back and have a look at the Kasparian strip. And this is a waterproof strip that's going to block the apoplastic pathway. This means that water can no longer travel through the cell walls and instead is forced into the symplastic pathway, where then it will pass through the cytoplasm of adjacent cells through plasmodesma. So if we look here, this was our apoplastic pathway. The water's being sucked through the cellular cell wall of your plant cells. And then you hit the Kasparian strip, which is waterproof, so it can't move through the cell wall anymore. It gets bumped into the cell, through the cell wall into the cytoplasm, and then it will pass through the cytoplasm between cells until we hit the xylem. So let's have a look at xylem in more detail. So here's 3 different pictures down to microscopes. Some of them are 3D and show the internal structures really nicely, and others are like a slice, a cross-section through a stem to show the vascular bundle. And the function of xylem is to transport water and also the little mineral ions that are dissolved in the water up the plant eventually until you get to the leaves. So we mentioned that they have this spiral structure, that they have a really distinct shape, and this is a material called lignin that we'll look at in a bit. What we need to know about xylem vessels is, first off, they're non-living. They're hollow tubes, they're the outside of cells, there's no internal components, they're hollow, they have no cytoplasm, they have no end walls that are open at both ends, so that if you had one dead cell here, let's say, there isn't a plate blocking the way. So instead, they're a hollow network of tubes that allow the water to flow up continuously. If you go back a little bit, you notice that our xylem vessels is not just one big straw-like tube, there's usually lots of them together, so they have little pits connecting them so that water can flow between different xylem tubes. And they are so strong that they do not collapse under the tension of moving water from the root up to the leaf. And that's to do with that spiral of lignin that we talked about. Lignin is going to hold the microfibrils in the cell wall together, or make them parallel, so they lie flat and nicely together and are nice and strong. And it also has a function in making the cell walls waterproof. When we look at transpiration, this is the whole process from the bottom to the top. So there's a really nice animation on the right hand side here where you see that water is leaving the leaf at the top and water is being drawn up the stem from the leaves in a continuous column. And this force that's making water travel all the way from the roots, sometimes several meters above the ground in a tree, like gravity defying, is called transpiration pull, which is the movement of water up a plant from the roots to the leaves against gravity. And there's a few forces that can drive this. First off, we need to start that pull up the top by removing some water from the top, because water is going to naturally be diffusing out of the mesophyll cells into an airspace, out through stomata and in the air. And then bringing this water from the roots up that trunk is the forces of cohesion and adhesion. So because we have the cohesion involved from water and the tension as it's moving up as a continuous column, the theory of movement is called the cohesion tension theory. Let's have a look at the way water moves. We'll take a moment to pause and remind yourself what is cohesion and what is adhesion. Use the labels on this diagram to help you. Let's look at our first definition. Because we know that water molecules form hydrogen bonds with other molecules, this means that they have a sort of stickiness to each other, they're attracted to each other. And we know that the big red thing here is our oxygen, the two little whiter things are called hydrogens, and the oxygen has a more negative partial charge, whereas the hydrogens have a positive partial charge. So naturally opposites will attract, and water can therefore be attracted to each other and move as a column up the xylem. So we can see here the cohesion is that force between different water molecules. And if we have a look at this little GIF, you can see they've imagined those hydrogen bonds as actual physical things here. You can see the little white lines, and they're all sort of attracted to each other, moving up together. Next is adhesion. You might have heard of it in terms of glue before. So think of it about water molecules interacting with something else. You're sticking different things together. In this case, it's water molecules with the walls of the xylem. And because they're doing this, because of the forces of adhesion, the column of water remains unbroken, and it just helps pull the water up together. So we've talked about a lot of different key processes so far. Take a moment to pause and think of as many key terms as you could possibly need to explain the movement of water through a plant so far. Let's have a look at our list. We've got water potential. You need to talk about the leaves. You need to talk about water, Xylem vessels, the process of osmosis, going through the stem. We need to know that if we're in the stem, there is pull or tension. Moving water is a continuous column with the aid of two forces, cohesion in its hydrogen bonds and adhesion in the process of transpiration. Take a moment to have a read of this. You should have it on your handouts if you're on my class. So have a look at all of those processes that are jumbled up in the wrong order. I'll have given you a link to a different video or a textbook page, your preference, and I'd like you to sort these statements out into the correct order. Let's look through our answers. So we're going to start with the water being lost from mesophyll cells via evaporation. We're going to end with the pull of water up the xylem creating a tension, so we have the term cohesion tension theory. So after we lose water by evaporation in the leaves, the water potential in the airspace becomes lower than the water potential in mesophyll cells. This means that water is going to be moving down a water potential gradient from the neighbouring, the next door mesophyll cell to the cell that's lost water. The water potential of the neighbouring cell becomes lower than the water potential in the xylem, and this means that water is going to again move down the water potential gradient from the xylem to neighbouring cells across the mesophyll. So think of it like a chain reaction. If one cell has lower water potential, let's say to make it easier to think about, it becomes more dry, then the cell touching it next door is going to donate a bit of water. And then that cell becomes a bit drier. So then the cell next to that second cell is going to suck a bit of water out. So you get this chain of cells together that are becoming, let me show you a little drawing. So we have all of these cells together, and we have a chain reaction occurring where this cell on the end has a very low water potential. So a little bit of water moves through from this one, and then a little bit of water moves through from that one, until you end up with this network of mesophyll neighbouring cells across the mesophyll, all set up in this water potential chain. And this is the process of transpiration, where water is drawn out the xylem. Let's pop our Xylem vessels in now. And it's going to be giving water to those mesophyll cells. The hydrogen bonds between water molecules cause them to stick together. This is called cohesion, and cohesion between the water molecules means that water travels up the xylem in a continuous unbroken column, so that is transpiration pull. So if you imagine that all of this is happening in a leaf, so these are the cells in our leaf, taking it from the xylem vessels in a vein. Let's imagine the xylem vessels running from that leaf all the way down the stem to the roots. And because this cell has the lowest water potential, pulling a bit more out behind it, all across the mesophyll in the leaf, all the way to the xylem, and then the water molecules in the xylem are drawing up more water through the xylem in the stem, which is connected to the xylem in the roots. So you get this continuous unbroken column. So for now, let's look at a few ways of putting this in words. From the root, water is transported upwards through the stem. Explain how evaporation from the leaves can cause water to move upwards. This is all in the process that you should be able to remember the name of. Take a moment to flick back through your notes and give it a go. So the first thing that we noticed before causing all of this movement to happen in the leaf was that the water potential in the leaf cells decreased or became more negative. And this sucked some more water out. It transported water out of the xylem into the surrounding tissues by osmosis. And as they were pulling along the chain, this is creating a pull or a tension on water in the xylem. and we know that all of that water is connected, so it's going to move as a continuous column all through the forces of cohesion due to hydrogen bonding, means that the column of water doesn't break due to adhesion with the xylem walls. So as a quick summary for ourselves, transpiration is to do with all the different bits of the plant. We have in the leaves, we've got transpiration or evaporation, where water is diffusing out from the xylem to the mesophyll and then out through the airspace through the stomata. In the stem of the plant, we have those xylem vessels, so we know about the cohesion tension theory. We know about the cohesion between adjacent molecules of water. We also know about the adhesion between the water molecules and the sides of the xylem vessel. And we know about in the roots, we're continuously drawing up more water because through osmosis there's a water potential gradient established favouring the movement of water into the root cells. And this is maintained by actively transporting different solutes into the root cells from the soil, which then will travel up the xylem into the mesophyll cells. Let's have a go at a few exam questions. We'll look at our first one together. We've got mineral ions such as calcium, nitrate and magnesium are transported in the xylem. They are also dissolved in the water. You need to describe first off how the structure of xylem vessels allows them to transport water. So how does the structure of xylem help its function? And it's a description question, so you're just stating some facts. For 2 marks, give it a go. Looking through our answers, the first thing that we can say is about how it's structured in order to be able to transport. How does water even get in there? And it can happen because it's hollow, there is no cytoplasm or there's no internal organelles. You could say that their vessels have no end walls or open at the ends, so they're more like a tube. You can also say that the vessels are really strong so they don't collapse, or that they have pits. For the same mark point, you could say they're strong to keep open. And you could also mention wonderful lignin making the walls waterproof. Let's look at the next question. We need to explain what is meant by the term tissue, because we know that xylem is a tissue in a plant. So, we need 2 marks for this one too. Well, firstly, a tissue is definition includes that it's a group of many or several cells. And the second part of our simple definition to remember is that they work together for a common function. The next one's a bit more tricky. We need to look at a few things here. So the first thing that we have to do is suggest how lignin adds strength to the xylem tissue. So here are some pictures showing some xylem vessels before you remove lignin, and we also have a picture of some xylem vessels after you've removed lignin. So you're comparing those 2 structures. We need to link this back and think, how does lignin add strength to xylem tissue? What evidence do I have in this picture that says that this photo looks more stable and it's more strong? And what evidence do I have from this picture that says that it's weaker or it's less strong? Let's look at our 2 things. The first part is relying on us to remember that cellulose cell walls are made up of microfibrils. So lignin is clearly holding those fibres together very well over here, but not so well over on this side. And if we have a look, we can follow those fibres across, and we notice that all of the fibres are running in the same direction on this side, whereas being a bit of a tangly mess on the second. So we can say that lignin fibres keeps the microfibrils parallel. Without the microfibers, the strands do not run parallel. Let's do a bit of math, shall we? The next one is showing us the concentrations of some inorganic ions as measured in soil and plants. So you have a table showing you from the leaf all the way down to the soil what is the concentration in parts per million of nitrate ions. What we have to do is calculate the percentage decrease in the nitrate ion concentration from the root to the leaf. So, between the root and the leaf, what is the difference in terms of percentage decrease? Let's look at how we'd work this out. Firstly, we need to make sure that we've got the difference in the right place. So before we had our two numbers, 2100 and 250. So if I want to do the change in between the two, you have to do 2100 minus 250, and you should get this as your correct answer for the top part. The next step is that you need to work out the overall percentage decrease. So we're going to base this on that higher number, so that was the original value before we did the difference. So I would have 1850 divided by 2100 to get our decrease value, and then we would times it by 100 to make it the percentage, so you could have anything in the region of 88% either expressed to 3 significant figures or to 4. Next up, you have to describe how nitrite ions are transported from the root to the leaves. So again, another quick 2 marker. What process allows this to happen? Describe how it works. First off, nitrite ions, even though this isn't the main focus of our lesson today, we know that those are dissolved in water, and those nitrite ions dissolved in water are going to be moving through Xylem vessels. Secondly, points for saying in water or through the transpiration stream, but we do not chat about active transport. That would only be letting us talk about how we're moving water into the roots, not necessarily all the way up to the leaves. And our final question. Plant cell walls contain cellulose. Cellulose molecules are polymers. First thing that you need to do is name the monomer, the single unit that makes up cellulose. Next up, you need to describe which of the following correctly describes how these monomers are held together in a cellulose molecule. So which bonds, and are they branched or unbranched? And lastly, we have cellulose molecules are held together in bundles called microfibrils. These microfibrils are embedded in a matrix containing calcium pectate. Calcium pectate can be found in which organelle in a plant? Ameloplast, chloroplast, middle lamella, or tonoplast? Let's do our final round of answers. So the first one, the monomer, the single unit that makes up the chain of cellulose as a polymer, is beta-glucose. You could also write it as the fancy B or just a little b glucose. The next one, our answer is B. Glycosidic bonds are in an unbranched chain. And lastly, in calcium pectate, that can be found in C, the middle lamella. So if you're in my class, remember you can email me at any time if you've got any problems about this. Have a good day, and we'll look forward to seeing you later. Bye bye!